couple minutes out here at the beginning of the class to talk to you about your lab reports that are due on Tuesday. When you come to lab on Tuesday morning at 7:45, you need to have two hard copies of your lab report. They need to be double spaced. We don't tell you to double space them because we're just frivolous wasters of paper. We want them double spaced so that we have space to write on them the comments that we write on them. We have space to fit that stuff in there. So adhere to the formatting that we asked you to do that. I've seen a couple of people's drafts now of results. Your results section should be written as a narrative. There's a narrative to it, and then your graphs support that narrative. The results section isn't statement, graph, statement, graph, statement, graph, which is oftentimes how results come to us at this stage. Review how to write out captions, where they're placed, how they're formatted, the information that is contained in them. I'm going to show you some figures in the course of lecture today, and so we will we'll visit that, and I'll pause for a minute and explain kind of what the figure is about, and then formatting your literature cited sections correctly. So these are just things that I've seen as I've looked at a couple of people's early attempts at finishing the entire lab report. If you go to the Moodle site, here's the Moodle site. If you go to the section called Lab-Related Materials, there is a section called Lab Report Materials, and in that folder is everything that you need to know in order to do this. Now, the cool thing about Moodle is I can get activity reports. This folder has been accessed 26 times by 11 users, but in that folder are multiple files. So it should be getting a lot more usage than it's currently getting, because inside that folder are all these things. And if you download the lab report overview or the lab report grading rubric, what you will get when you do that is you will get, in this case, the lab report overview. And when you look at this, it basically has this thing all broken down for you in grinding detail. So those of you who haven't seen this, you need to go and read this document and pay attention to things that are in it. It gives you formatting instructions, double space, Times New Roman font. You don't need a title page, just put your title at the top of the first page and then write. Tells you what we expect to see in the introduction, how many primary sources you need to have, et cetera, et cetera. Under literature cited, it gives you examples of how to do in-text citations appropriately, how to do things when there are more than 3 authors. It also shows you how to put a literature cited in your literature cited section. Notice that all lines after the first line have a hanging indent. That isn't hanging out there like randomly. We want you to do it that way because that's the way they're usually done. There's a hanging indent thing. It's right up here. To get a hanging indent, all you have to do is go up here and grab that little triangle and drag it over a 1/4 of an inch or 1/2 inch or whatever your hanging indent is going to be. You'll notice that that's the hanging indent that is doing this. If you do that at the beginning of your literature cited, then every literature cited entry that you put in there will automatically do it because you'll just be continuing to type that hanging indent in there. So everything you need to know about how you want to see the thing is in this document. Go and read it and pay attention to the things that are in it. You will ignore them at your peril. The reason that you will ignore them at your peril is that we have a rubric, which is also on the Moodle site and has been for a little while now. And you will notice that in this rubric, under overall impression, proper formatting of the document as a whole is part of the thing that we're going to be grading you on. Formatting the literature cited appropriately is one of the things that we're going to be grading you on. We're going to be grading you on a bunch of other things as well, like is your introduction structured in a way that makes logical sense? Does it cover the literature that pertains to your topic adequately? Does it set up the reader with an understanding of what the general issues are that your project is about, so that when we get down to the hypothesis and your predictions and things like that, we understand why that makes sense in light of what's known about this topic as a whole, et cetera, et cetera, et cetera. You can go and look at the rubric right now and see how much is the introduction weighted, how much are the methods weighted, how much are the results weighted, et cetera, et cetera, et cetera. What are the things that they're going to be looking at as they go through and read my lab report? What are the kinds of things that I need to be paying attention to? This is the rubric that sits next to me when I sit down with your lab report and grade it. So there's information in there that's useful to you. Please, please, please go and read it between
just to
now and then.
elaborate on that a little bit. All the material you're going to write about, your introduction, your methods, your discussion, that's all information you all have learned, you're synthesizing, and you're earning those points. Think about all the formatting stuff that you just presented as sort of free points. We're not just making that up for a reason. Paul and I and all scientists have to deal with those requirements from journals all the time in order to publish.
There are formatting instructions at every journal.
Every journal has their own, and so we've picked a series of representative formatting instructions for this manuscript, or for this lab report. But that's a realistic part of being a scientist, and if you publish anything during your junior and sophomore year, or as a graduate student, or professionally, you'll have to go through all this kind of stuff.
Yeah, and we chose a format, one, because it's an ecological journal format, and two, it's one of the simpler formats to adhere to. It's really simple: authors, date, article title, journal title, volume number, colon, page range. It's a really pretty simple format to master, so it's not difficult. Just be consistent, do it, and do it consistently as you go through. So does somebody else have a question? Yeah.
I was, when I was going through and citing things, I was citing some of my literature didn't have page numbers.
Okay, so it's because you're getting digital versions of things, and oftentimes a digital version of something will just have a DOI. I don't know what DOI stands for, but it's a DOI address, and it's an address that sends you to some digital version of that. If that thing has been published in a journal somewhere, you should be able to type in the author name and the title name,
Okay.
and it should take you to the journal's website, and you can get the date it was published, the volume number that it was published, the page range, all that
Okay.
stuff. It's available. It's just that you found it through something. I noticed on your drafts, there was a couple of you that had a bunch of things that were citable. Well, Citable isn't actually a journal. Citable is a service that Nature has. So it's a website at Nature, which is a journal, a series of journals actually, where they have a bunch of papers, digital versions of papers that are citable, except that they spell citable this way, strangely enough. And so it's just calling attention to papers that they think would be interested to a broader topic. But Citable isn't a journal. Citable is a website where they've done this, but those papers are papers that are appearing in either Nature or Nature Ecology and Evolution or Nature Some Molecular Biology or whatever all the Nature journals are. And so there is somewhere at Nature the citation information for that article, but you're getting it off of not the articles, not the journal's website. You're getting it off of some accessory website. In the same way that when you go to a Semantic Scholar, some of you cited Semantic Scholar. Semantic Scholar is just a repository for where people put their papers, digital copies of their papers, but the paper actually came from a journal somewhere. You need to find that journal and the citation material, the citation information for that journal. What I should be able to do in a literature cited section is I should be able to easily locate that article either in a library or online or something else. And the way you do that is by giving us the authors, the date, the article title, the journal title, the volume number that it appeared in that journal, and the page range that you can find that actual article.
You can usually find that information by Googling if you have an author or the title or the year or some combination of those. You can also search the digital object identifier number, that's what DOI stands for, and and that'll often pull up the paper directly.
Yeah, so going back to this. I did not put the journal name in there, but let's see. Well, the first thing that goes to is Dan Sign's website at the U.S. Forest Service. And so this is the Forest Service repository of where articles have come out where Forest Service employees are co-authors. And once again, it has all of the authors. It has the date, it has the journal, it has the volume number, it has the page number. You could put this into Google Scholar and search, and it would also return the same information. It's out there and it's available. You just have to spend 20 seconds per article making sure that you've dug it up.
Does everyone know the difference between Google and Google Scholar? If you had done the same thing on Google Scholar, it probably would have gone to the paper.
Let's do the same thing on Google Scholar. I just put in the authors, the date, and the title. Oh, and that is so weird that it's the USDA Forest Service is the only place that it appears. Because the Journal of Herpetology has a website. I don't know why it's not coming up.
It's just because Google is seeing the Forest Service website, and that's why I offered that PDF.
Oh, and so here it is at the journal. So this is the Journal of Herpetology. Once again, here's the title. Here are the authors. That's the journal. There's the volume. There's the page range, et cetera, et cetera. The other thing that you have available to you, don't forget, we have a library website as well. And you can go onto the library website and once again go to databases. Databases. I mean, my hope is that you've been using these thus far, and you can go to, for whatever reason, ours are found in General Sciences. Go down to Web of Science. And since Dan Signs is the person that everybody seems to know from this paper irritatingly, even though he's not the primary author, we can type in the things that are in the title. And hopefully, when we do this, we will get Dan Sign's master's thesis, but we also get comparison of dietary overlap between Alan Patrick and Sid Patrick Dekos. Klinsky, Vaughn, Signs, et al. Will Godwin didn't make it. Journal of Herpetology, volume 28, pages 225 to 230. We've now skinned this cat 4 different ways. This is an easily skinnable cat. So you have resources available to use those resources. I don't get so caught up. What's so funny, Nathan?
I think some people aren't used to the many ways to skin a cat metaphor.
Oh, I'm sorry. You guys have never skinned a cat? Have you heard the phrase?
I've heard
There's
the phrase,
more than
yeah.
one way to skin a cat.
No.
Oh, that's a phrase. I don't know where it came from.
It's
But
an
it-
idiom I'm pretty.
But it basically, there are generally many ways to do a single thing. I don't really care how you get the cat skinned, but I want to skin a cat on Tuesday morning. Sorry.
It's our next research project.
Sorry. Don't get so caught up in formatting that you neglect the really truly important things, which is content. Be sure that you're reviewing the literature adequately. Be sure that you are structuring your introduction in a sensible way that makes sense to you, the writer, and me, the reader. Make sure that you reflect on the introduction in your discussion as you're discussing the importance of your results, not only with relevance to your hypothesis, but also what do your results say about all that literature that you reviewed in your introduction? Because what you find relates not only to what your particular question was, but it also relates back to this broader body of information. In these in these lab reports, typically, introductions are too short, discussions are too short, and also, oftentimes the results sections are just weird, because it sometimes feels like you don't have a lot of results to share. In the butterfly group, actually, you have quite a few results to share, and I'm not sure what the other group has to share yet, but make sure that you're writing that out as an area. We're gonna see some results section stuff today as we go through today's material. What was it that I asked you to remind me about? Oh, yeah. What were you about to say, Nathan?
I think I have it written down here. I'm gonna turn my pages to.
Okay.
So go ahead.
There is a student named Riley Martin who needs human subjects for her senior research project. She asked me if I would offer extra credit to students who are willing to participate. So I talked it over with Dr. Judd. We're willing to add 5 points to your final exam if you participate in Riley's project. She sent me earlier today a write-up of it with a link on how to participate and all that stuff. So if you're interested in that, she'll keep track of who you are, and then she'll send us the names of who's participating and who hasn't, and we'll just tack 5 points onto your final exam if you want to do that. She said it takes, somebody in here has already done it, right? How long does it take?
20 minutes.
Yeah,
20 minutes.
20-ish minutes.
So-
It's really not that. It's basically nothing.
Was that the thing that I asked you?
Yeah.
Okay, all right, cool. I asked Dr. Judge to remind me of things that I will inevitably forget. Okay, any questions about lab report? 2 copies, Tuesday morning. Questions? All right, the next thing that I want to talk to you about is the schedule, or the schedule, if you're British. We're approaching the end. Congratulations on making it this far. So today we're going to talk about this chapter that you read. On Monday we're going to talk about statistics. On Wednesday we're going to talk about quantitative genetics. On Friday, Dr. Judd will talk to you about adaptive landscapes. And then the day before you leave for Thanksgiving, you will have your last literature summary due. So your last literature summary is Brody and Brody, 1990. It is in the Moodle site during that week, so it's down there. So don't forget to write a literature summary for the day before we leave for Thanksgiving. The other thing is, you've noticed that for the Veen et al. Paper and for the Hendry et al. Paper, Dr. Judd and I came to class prepared with a set of questions that would hopefully walk you through your discussion with your peers of those papers, because we're trying to develop in you the ability to read scientific literature critically. Your other assignment for that day is you need to come into class with a set of questions that you think would help walk your peers through your treatment of that paper as well. So you have 2 things to do that day. One is to come with a literature summary prepared, and the other thing is to come with a set of discussion questions that you will use in your groups to discuss the paper in the same way that we've given you questions on papers before. Now, the rest of the semester is based in reading papers. So after Thanksgiving, you're going to basically read a paper a day. You're going to read Brody and Brody 1999. You're going to read Gaffney 2005. You're going to read Hannafin 2008. You do not have to write literature summaries for those things. But what you should do for each of those, your preparation homework in preparation for each of those is 1, read the paper, and 2, once again, come to class with a set of discussion questions that you think will be beneficial for your group discussion. Because the whole last week of classes is going to be us coming to class and discussing primary literature that kind of walks us through this system. Yeah, Javon?
How many questions do we have to write for the Brody and Brody?
Enough to cover the paper. So the last time I did this for Beam, there was a lot of questions. So you want to cover the paper adequately. Dr. Shrek?
Just try to look at what the questions about the other 2 papers have in common with
Sure.
each other.
Use that as a guide.
Kind of a structure or guide to the questions you're going to write.
The nice thing about having all of you doing this is everybody's going to come up with different questions. And between the 4 or 5 of you in each group, you'll have plenty of questions to kind of guide you through the discussion. On each day, we're gonna do what we've always done, which is pause at the end of class and do a recap to make sure that everybody in the class has the same understanding of what material was in the paper. But anyway, just get ready for reading literature. All of these articles are already up on Moodle, so you can download them and have access to them as soon as you want them. As a result, while we'll have lab next week, we will not have lab the week after that, even though there are classes. Well, there are classes on Tuesday.
Please give us that.
No, you already said we were having class.
There is technically no lab scheduled. If for some reason you need to meet with me to get ready for your oral presentation, which is Tuesday as soon as you come back from Thanksgiving. So the semester is shortened, so normally we would have 2 weeks of meeting after Thanksgiving. This year, this semester, we go away for Thanksgiving, we come back, and that's the last week of classes. So that Tuesday, you have to make your oral presentations. So you miss a week of preparation, essentially, when you could have come back from Thanksgiving and met with your group to get your oral presentation ready. So if you need to meet with your group on Tuesday, if you need to meet with me on Tuesday to help you work on your oral presentation, use that time that you have available. There is no formal lab meeting scheduled the week of Thanksgiving. So, but I'm around if needed because I live here and I'm not going anywhere.
Cool.
Sad but true. Questions about the schedule? All right. Well, congratulations on making it to the last case study. This is, in my mind, the funnest case study. For a variety of reasons, I just think it's kind of fascinating. It's a case study having to do with a species of newt that lives in the Pacific Northwest and a species of snake that eats it. And so it's a predator-prey interaction case study. So the goals for today is to understand this thing called the predation sequence and how predators and prey deal with one another in predator-prey interactions, and also to gain some insight into the process of science by looking at the book chapter that I asked you to read. Before this book was published in 2011, we had the students read a summary article that appeared in the journal Bioscience that summarized some of this. But this book this book chapter that we give you, the book itself is designed to be read by lay people, not scientists. The audience for that book is not a scientific audience, and it also covers a much broader time frame for the research than the earlier paper that we had people read. So when this book chapter came out, we made this the introductory piece of literature to get you into the process. And it basically reviews all of the literature that you're going to be reading for the rest of the semester on this case study. So it serves as a nice introduction. So first, I want to impress upon you the importance of predator-prey interactions. This was a paper that I read back in graduate school. During any given day, an animal may fail to obtain a meal and go hungry, or may fail to obtain matings and thus realize no reproductive success. But in the long term, the day's shortcomings may have minimal influence on lifetime fitness. Few failures, however, are as unforgiving as the failure to avoid a predator. Being killed greatly decreases future fitness. If you're dead, you're not mating. That's it. Your genes are gone from the gene pool. And so this is actually the first paragraph of a paper written about behavioral responses of prey to the threat of a predator and how prey deal with this. So I reordered this and I reordered it in the wrong way. Or did I? Oh yeah. So here it says, predation may thus be a strong selective force over evolutionary time, and has long been recognized as important in the evolution of adaptations such as cryptic and plasmatic coloration, et cetera, et cetera. So I would like to know from you is, like, when you hear the term adaptation, what do you think? Siobhan?
The way that something evolves over time due to things around it or surrounding it.
Okay, so is adaptation a verb or a noun?
Yes.
so if it's a noun, how would you define adaptation? What is an adaptation? The previous slide had some examples. Crypsis is an adaptation.
Okay, so I guess let's say I'm not sure.
So if it's a noun, so what would be a noun that would describe an adaptation? So if something is cryptic, what is it?
I don't know.
It's not secretive. So who knows what crypsis is? Hmm? Yeah, it's camouflage. If something is camouflage, so camouflage in this case is what? What would you describe camouflage as? So it's an adaptation, but describe what, if you were telling your mom what camouflage is. It's a trait. In this case, camouflage is a color pattern, and so we can put this down here, a color pattern. But what makes that color pattern an adaptation? Javon? You start down this path, I want to continue with you. The path.
Okay, so I guess
What does this trait impart to an individual?
it's like, you say in part?
Yeah, what is it? What characteristics or qualities does this give to the animal possessing that trait? Okay, in this case, it helps you hide because that then helps you what?
Survive.
So it helps you survive, and survival is a part of fitness. So in reality, an adaptation is a trait that does what? In terms of fitness.
Increases
The trait. Does anybody else have another definition of adaptation? So this is adaptation as a noun. Can adaptation also be a verb? Yeah. Now what would be a verbal definition of adaptation? Jones.
A process by which traits are gained or lost in response to evolutionary pressure.
Anybody want to critique that definition? Oh, gain or loss what? By?
In response to evolutionary pressure.
Lucy, do I see your hand up?
Yeah.
Take it
Instead of
to-
saying traits are gained or lost, evolved in response to selective pressures,
What's the problem with gained or lost?
it's saying, it's kind of implying that it comes in one generation.
See, there's a, if something's lost, how can not having something be an adaptation? Is one thing from a strictly logical point. And certainly we can be a little better with our use of terminology. Traits evolve. So it is this process. But at the heart of this process is you end up, the end product of this process is that you have something that someone might call an adaptation. That is presumably some sort of trait. It could be a morphological trait. One group, the water bug group, is studying a morphological trait in their water bugs. But that morphological trait could contribute to fitness. It can be a behavioral trait. It could be a physiological trait. There are lots of different kinds of traits that you can use. Let's look at a section of literature where people wrote an entire paper about what an adaptation is. In evolutionary biology, we encounter two different meanings and a possible conflation of concepts for features called adaptations. The first is consistent with the vernacular uses cited above. A feature is an adaptation only if it was built by natural selection for the function it now performs. The second defines adaptation as in a static or immediate way as any feature that enhances current fitness regardless of its historical origin. So these two people who wrote this paper would say, for an adaptation to be an adaptation, it not only has to be a trait that contributes to increasing fitness, but it has to be a trait that evolved to do its current function. And they're making what might seem like a little petty and fine-scaled distinction, but organisms are really good at co-opting things. Something will be evolved in response to selective pressures for essentially making you better at one particular challenge, and then over time, those things get co-opted for other purposes. So for example, I normally bring my phone today. Does anybody have a phone that I can borrow for just a minute? I forgot my phone. So let's say this is my phone, and I'm gonna text my girlfriend on Jackson's phone. What am I using? I'm using my opposable thumbs. Now, I don't mean to be crude, but is using my thumbs gonna get lucky?
Not necessarily.
Is using my thumbs to text my girlfriend going to increase my fitness potentially? Sure.
Yeah.
Hey, what you doing tonight? So, can having opposable thumbs increase fitness?
Yeah.
Yeah, potentially. Probably if you're somebody other than me. Did opposable thumbs evolve to enable texting that might or might not increase our fitness?
Of course not.
Of course not. We had opposable thumbs long before we had phones. So even though we have opposable thumbs and they might have evolved for one purpose, that doesn't mean that later on they can't have a different purpose that might contribute to increasing fitness. And so it's an important distinction, and it's an especially important distinction in this particular case study, because in the chapter that you read, Butch Brodie, the younger Butch Brodie III, makes the point about the evolution of toxicity in these newts. And he talks about whether this toxicity evolved in response to predation by snakes, and whether snakes' resistance evolved in response to newt toxicity. And how can you go about telling those two things apart? What was the evolutionary pressure, the selection pressure that led to the evolution of toxicity in the newts to begin with, and what was the selective pressure that led to resistance to tetrodotoxin in the snakes? That is sometimes a difficult question to tease apart. We refer to toxicity in the newt and resistance in the snakes as adaptations, and we make the underlying assumption that those two things evolved in response to selection pressure being exerted by each of those two species, but it's not necessarily a given. And so when you think about adaptation, think about adaptations as traits that increase fitness that should, in theory, have been evolved in response to selection to do the current function that they currently accomplish. So predator-prey interactions are complex in the thick of things, and so two guys, Krebs and Davies originally, but now Davies et al., have a textbook that I use in my animal behavior class, and these are the guys that first got me on the path of taking complicated processes, complicated information, and busting them up into tables. So when you think about the predation sequence, they find it useful to think about what a predator does while it's trying to go about catching prey and eating prey and things like this. And so they've divided the predation sequence up into 4 phases that represents the predation sequence, but these 4 phases are kind of named after the predator activity. What are the things that a predator has to do if it's going to get itself fed ultimately?
Find thing to predate.
Okay, so you have to go searching for food. So in the search phase, there are going to be things that you have as a predator, adaptations that help you search, and there are things that prey have that help them avoid becoming prey. Jackson, what would be the next phase after you've searched for food?
Like, once you've like found, like after your search and you found it, you have to enact on actually hunting the prey down.
Okay, but before that, so that would be what? What would be a good single word for that?
Trying to think, searching.
When you stop searching.
When you stop searching, actually, you're two steps ahead of the next thing, actually.
Okay.
But yours is a valid phase in it. So you
Why
search
are-
for it.
Why are your keys always in the last place you found them? Or the last place you looked? Why are your keys always in the last place you looked?
Oh, because I'm an idiot. That's why. That's why that's the case for me.
Confirmation bias?
Because you found
Jonas is
it.
on to it. Why are your keys always in the last place that you look?
Because then you find it.
Because that's when you find them. So
Stop looking.
So that kind of gets at it. If I lost my keys, let's say that I lost my keys in this room, well, keys look like keys, unfortunately. Let's say that I lost a scarf in a room full of scarves. How are you going to know which scarf is my scarf if I recruit you to help me find my scarf? Okay, so if there is a room full of plaid scarves and spotted scarves and scarves made from mohair and scarves made from camel hair, my scarf is the hand woven from what's the name of that yarn that my scarf is made out of? Oh, it's bamboo. It's actually woven bamboo. Mine is a hand woven bamboo scarf that is kind of a powdery blue color. Now when you go looking for the scarf in a room full of scarves, what do you know that you're looking for? A blue bamboo scarf, right? So that when you find my scarf, you will
Stop searching.
stop searching because you recognize my scarf as the scarf that it is. And so we search for prey
Until you recognize prey.
until we recognize the prey. An organism will run across all kinds of things that it could potentially put in its mouth and eat, but some of those are going to be prey, and some of those are going to be things that you really don't want to put inside your mouth. And so you have to be capable of recognizing the thing that you see as prey. And there are things about the predator that enables them to be better at recognition, and there are things about prey that keep them from being recognized as food. Can you think of something that keeps you from being recognized as food?
Smell.
Camouflage.
Camouflage might keep you from being recognized as food. Then what's the next thing you have to do, Jackson?
Smell.
Capture.
You have to capture it. So the next phase is capture. And then is capturing something all you have to do?
No.
You have to kill it and eat it. You have to handle it. And so we have these 4 phases of the predation sequence, and it's a useful way of thinking about things because at each of these phases, the predators have adaptations that help them procure prey, and the prey have adaptations, counter adaptations, that help them get away from the predator. Search, recognition, capture, and handling. This table continues to basically say, okay, in searching for prey, what kinds of things are useful? Improve visual acuity, having a search image. I took a group of students from Texas A&M out to a marsh when I was in graduate school to search for salamanders. And the salamanders that we were searching for are green, they're elongate, they have thin bodies, they have a blade-like tail. They look a lot like grass. And in this marsh was a bunch of grass also growing in this marsh in February down in East Texas. And this was a marsh that we took the herpetology students to on a regular basis. Me and Dan Sines, who gets all the credit for writing the first paper that we ever wrote together, spent a lot of time at this marsh in the early spring looking for salamanders and frogs. And when we took these guys from Texas A&M out to this marsh, and they were all good herpetologists, very good at catching amphibians and reptiles, they were super frustrated because we were finding all of these salamanders and they were finding nothing. And we didn't necessarily really like these guys, so we kind of rubbed their noses in the fact that they suck so bad at this. But in the end, what we told them is the only difference between you guys and us is we've spent a lot of time in this marsh such that we've developed an eye, we've developed a search image for how to recognize these grass-like salamanders in a pool full of grass. If you spend as much time out here as we have spent out here, you would be finding the same number of salamanders that we have because you will have developed a search image. You know what a salamander looks like against a background of grass, and that then enables you to find the thing that you're looking for better. So it improves your searching. That's what they're talking about when we talk about a search image. Organisms can counter those adaptations by doing things like crypsis and polymorphisms, spreading themselves out so that predators come into contact with you less often, so the predators have a more difficult time developing a search image. Any of you guys ever seen the movie Shooter? In Shooter, the last episode in the movie, the last action episode in the movie, Mark Wahlberg is on the snow in a ghillie suit. A ghillie suit is a suit that is designed to break up the outline of your body so that there aren't any hard edges that would cue your eye into seeing this. So when we talk about disruptive patterns, that's not only is it background matching, but it's also disrupting the outline of your body. Recognition of prey requires learning on the part of the predator. You have to learn what the prey look like. This happens in things like aposematism, when you're faced with a bunch of coral snakes that have all kinds of different color patterns, and in those same environments, you're faced with a bunch of milk snakes that also have a variety of different patterns. If your goal is to eat a snake and not be bitten by a venomous snake, you have to worry about all of these coral pattern snakes, which means that you're probably going to also avoid all of these mimetic, non-venomous snakes. And so learning is required in recognizing prey. Very difficult for birds to tell the difference between coral snakes and non-venomous mimics of coral snakes. And so what they end up doing is they just avoid anything that's banded. And they eat unbanded snakes and striped snakes and things like that when they find themselves where there's a diversity of different color patterns. Catching prey. You've searched for the prey, you've found the prey and recognized that it is prey. Now you have to gain control over it. And so you can sneak up on them, you can have the speed that will allow you to track them down and catch them. You can have weapons of offense, claws, teeth, etc. But prey can also do all sorts of things to keep from being captured. Do any of you guys know what this behavior is called? I don't expect you to. You would learn it in animal behavior, but you might know it if you've watched a nature documentary. It's called stotting. It's this weird thing that gazelles do, and this is a gazelle from Africa, where they basically bounce on all four legs without really bending their legs at all. And it's not particularly good at running away from a predator, but they only do this when they've spotted a predator that is sufficiently far away. Normally, if a predator pounces on them, it like takes a secret of approaching, gets close to you, and then comes chasing at you, this gazelle would not be stotting, it would be running, haul-ass, getting away from the cheetah that's coming after it. But if the gazelle sees the predator far enough away, it will engage in this stotting behavior, and it's basically a signal to the predator that says what?
I've seen you.
I've
and I'm
seen
really
you.
good at my legs.
Nanny, nanny, boo-boo, you can't catch me because I'm aware of where you are, and I know that you're far enough away that if you start coming after me, I'm gonna be gone. And so don't even waste your time. It's basically a signal to predators that says, don't waste your time here. And it's a very stereotypical, every gazelle does it the same way. It's a signal that signals something to a predator. You can signal to a predator that it's been detected. That's what stotting is. You also have other things like the predator has weapons of offense. Well, prey can have weapons of defense. We've already talked about horned lizards having horns on their heads. We have the ornate box turtle here in Missouri that can close itself up in the shell. So even though you may have captured that individual, chances are you're not going to be killing him and eating him. And killing him and eating him is the next phase. So once you've got the box turtle, you have to be able to handle it in a way that you turn that box turtle from a captured box turtle into a box turtle that is in your belly. Now the box turtle has defenses against that, and presumably predators would have, once again, subduing skills that help them handle that. Box turtles actually, unless you're a really big predator, are actually pretty immune from being preyed upon once they reach a certain size, because they literally, their plastron hinges up, so you can't even fit a knife blade inside there, and they just are this hard ball of bone. But you can also have things like active defenses, like spines we saw in the horned lizards, and you can also have toxins. And this gets us to the topic today, which is toxins. Here is a rough-skinned newt from the Pacific Northwest. This is Turrica granulosa, the species that occurs there. What are some things that you notice about this newt? Noah?
It's colored.
Okay, so it's colored in a particular way. What's the nature of the coloration?
Kind of orange.
It's orangish on the bottom. What is it on the top?
Brown.
It's brown, and so it has a coloration that we know as counter shading, which means that you're darker on the top than you are on the bottom. What else do you notice? Elizabeth?
That's hard to color too, but it really stands out from the background.
Okay, so it actually kind of blends in with the background. It would
But
blend
the
in.
bottom side kind of stands out.
Bottom side kind of stands out, certainly. What else do you notice about the newt? Riley?
Has a really long tail.
Does have a really long tail, pretty long tail. Bronson?
It's got a small head comparatively to the body.
It has, well, yeah, maybe it's a little hard to tell from the angle that I've taken the picture at. Miriam?
Its eyes are pretty big compared to like its head
Eyes are
size.
pretty big. What else do you notice about it? Rachel?
It kind of has a rough skin.
It does have a rough skin, which is why it's called the rough skin newt. Lucy, why does it have rough skin?
There's a toxin in its skin.
Yeah, so those bumps are what? Luke?
Pores.
They're not pores, they are Javon.
Something
Go ahead.
to get it out, I guess. Creature gland.
Yeah, they're glands. So each of those little bumps is a gland, and the gland has a pore. What's that?
Sebaceous gland?
They're not sebaceous glands, actually. They're a type of gland called a granular gland. We don't have them in humans. They only occur in amphibians. So nothing that you would have learned from anatomy or histology or anything like that really has a corresponding type of gland. So this thing is covered from the tip of its nose all the way to the tip of its tail with these poison glands, these toxin glands that produce a particular type of toxin. What kind of toxin? Hmm?
Is it TTX?
TTX, which is shorthand for tetrodotoxin. And this is the toxin that these guys produce. And this individual is in the genus Turrica, and it's in the species Turrica granulosa. So Turrica granulosa is in a family of salamanders, the Salamandridae. And the Salamandridae is a family of newts. It is the family of newts, actually, and many of the newts secrete toxins just as part of what they do. And they have behaviors that are associated with this, and so these things do a thing called the Unken reflex. If you go and poke and prod on a newt, it will engage in this pattern where it basically pulls its head up over its back, pulls its tail up over its back. In some cases, it's just supporting itself on its belly and ribs, and it flashes this bright orange underside to whatever the predator is Why would you do something like that? Clearly, this newt cannot run away when it is doing this. Jackson?
It's the same principle that the poison dart frogs in the Amazon use.
Okay, so what do we call that kind of coloration?
I'm blanking right now.
Somebody else? Lucy? Aposemacism. So this is a posematic coloration. It's a bright color that signals to a potential predator, I'm nasty, you want nothing to do with me. And so if you flash this at a bird and a bird has never encountered a newt and then tries to eat the newt and then gets sick and almost dies, it's probably going to remember that bright orange color, and then when it sees bright orange color on salamander looking things, it's probably going to be like, been there, done that, I'm not going to do that again. And so it's a coloration that helps predators learn when you are toxic or dangerous. And this is just these various. This guy has done it so strongly, he's actually tipped himself over on his side because he can't hold himself up. So there are two Brodies. There's an older Brody, Brody Jr., who was department chair at the University of Texas at Arlington while I was there, which is one of the reasons that I know about this case study. And then there is his son, Brody III. And Brody III was a graduate student at about the same time that I was a graduate student. He was just getting out of graduate school, actually in the second year, I think, of my PhD program. And he tells you in this chapter about what he was doing in Oregon when he started thinking about all these things. What was Butch Brody III doing when he noticed that some of the snakes that he was collecting were eating newts rather than other things? What was he working on? Do you recall from the book chapter? Lucy?
I don't remember what he was originally trying to figure out, but I do remember that he was massaging the snakes to make them basically puke up what they had just eaten.
So he did a lot of palpating of a snake to get it to regurgitate its food, but he was actually studying this species of snake here, which is Thamnophis ordinoides. Thamnophis ordinoides is interesting because of what thing about it was interesting to Brody. Yeah.
It's the stripes versus squares
It's
one.
the striped versus checkered ones, and he was interested in the evolution of this polymorphism and why you would find striped snakes and checkered snakes in the same population. And so when we were talking about adaptive landscapes earlier in the semester, it's this guy's research that I was telling you about, where you have this correlation between anti-predator behavior and the morphology of the snake, such that striped snakes tend to run in a straight line and checkered snakes tend to reverse themselves. But while he was doing that, he would, what happens oftentimes when you catch a snake and put it in a bag to take it back to wherever you're taking them, is that that upsets them. They find the act of being captured stressful, and they will throw up their food in the bag. So you put you put a snake in the bag, and then when you get back to your camp, you open up the bag, and then there's a snake and everything that was in its gut. And so it's relatively easy to make snakes throw up. And in the same place that he was finding Thamnophis ordinoides, he was also finding this species, Thamnophis totalis, and when he noticed them throwing up and when he would count them, he would find that Thamnophis ordinoides was eating what mainly? Thamnophis ordinoides has a very narrow dietary preference.
You know, they were eating the newts.
Thamnophis totalis is eating newts. What does Thamnophis ordinoides eat? Yeah. Worms and slugs and things like this. So they have a very narrow preference of food. Thamnophis totalis, though, would eat what? It would eat newts, but it would also eat birds and mice and frogs and all kinds of things. And so what he noticed was that occasionally when he would catch a Thamnophis totalis and palp up its stomach contents, he would find that it would have newts in it. So this is just a reminder of you. This is what he went there for. And what he found was that some of these snakes were eating newts. When he saw a snake barf up a newt, what did he think about? What did that remind him of? Yeah.
There was an
So in the chapter that I asked you to read, it's about the parable of the coffee pot. Somebody other than Lucy tell me what that's about. Yeah, Rachel.
These guys were camping in the forest, and they were all found dead, and so they searched everything, and then they found there was this newt in the coffee pot. Apparently the newt had a poisonous strong enough to kill him.
Yes, and so he had heard this story from who?
His mom.
His father, because his father heard the story from who? Yeah, heard from one of his faculty members at the school where he was an undergraduate. And he was interested in doing research. He went to a professor, said, hey, I'm interested in doing a research. And the professor said, hey, I heard this story. Found these dead fishermen next to the stream. Apparently no signs of violence or anything else. But the only thing that they found that was out of the ordinary was that inside the coffee pot was a boiled newt. What don't you do for your research to find out if that newt is toxic? And so that's what Big Butch, Edmund D. Brody II, Edmund D. Brody Jr. went and did. It was got like a senior research project, and he ended up converting that into his master's degree thesis. And so these are the two Butches. This is Brody Jr., this is Brody III. They're called Little Butch and Big Butch simply because one is older than the other. Little Butch is this guy here who was at the University of Chicago. This is Big Butch, who was department chair where I was getting my PhD. And Big Butch wrote a paper in 1968, if I recall correctly. Yeah, 1968. I was 2 years old at the time. Basically looking at how toxic these newts were. And what he did was, and you didn't have this, I don't think this is well fleshed out in the chapter. He basically ground up this. Well, no, he did talk about this. He's basically given a mortar and pestle and a syringe and told to go find some newts, grind the skin down, create an injectable fluid that is the skin and all the toxins that are in the skin, and inject it in a ton of animals. These are all mammals from here down to here. This is some birds. This is Tarica granulosa itself. This is some snakes. And when you look at this, you see that the dosage is really, really tiny until you get to the newt, in which case it's way, 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 way higher. 0.0002 to compared to 0.2 and 0.4. And then this is the different concentrations and basically how long it took you to die. You can never do this kind of research now. The Institutional Animal Care and Use Committee would not let you do this. But what he noticed was that when he got to Thamnophis, when you get to Thamnophis sirtalis, you see that it takes a lot of toxin to knock them down and kill them. And this is 80 minutes, 36 hours, 68 hours, 100 minutes, 2 hours. Rather than it taking 5 or 10 or 15 minutes, it's now taking, in some cases, days for the animal to die, because presumably the animal is more resistant to the toxin. He gave it to mice at different concentrations. The more of the toxin, the faster the mouse died. He gave it to fish. He force-fed fish to see how long it took them to die. And once again, time to death in minutes. Fish are not immune to this toxin at all. So, Big Butch graduated, went on, got his PhD, and studied predator-prey interactions, but it was a while before he got back to the system. And he didn't actually get back to the system until his son was in graduate school. But there's some questions that can be asked. What are the ecological forces that push snakes to eat this poisonous prey? Why would you eat a prey item that could potentially kill you? The snakes evidently have some resistance, as you saw from that previous figure. Was this an accidental byproduct of garter snake physiology, or did these snakes evolve resistance in direct response to being in contact with these poisonous newts? What mechanisms enable garter snakes to resist a toxin that killed all of these other vertebrate predators, and were resistant snakes the evolutionary cause of extreme toxin in the newt? Did the toxin in the newt drive the evolution of resistance in the snake, and did resistance in the snake drive the evolution of supertoxicity of these newts? And these were the kinds of questions that sort of came out of knowing that there's this toxic prey item that some resistant predators eat. Well, as it turns out, garter snakes are really good for this kind of research, and one of the reasons that garter snakes might be prone to eat toxic prey is they have in many cases, very narrow dietary preferences. So this is Stephen Arnold. He is Little Butch's doctoral advisor, and he did this work back in the 1980s where he looked at chemoreception in garter snakes. This is a little baby garter snake, and that's a Q-tip. And he would swab Q-tips with, swab slugs with a Q-tip and get some slug goo on the Q-tip, and then he would present it to the snake. And when snakes are interested in something as food, their tongue flick rate goes up because they use their tongue with their sense of smell. They have a thing in the top of their mouth called the Jacobson's organ. They collect scent particles on their tongue, and then they wave them around in this Jacobson's organ that helps them smell. And so when they're interested in something, their tongue flick rate goes up. So you can just film them, count the number of tongue flicks, and that tells you if they're interested in it. These snakes lived on the coast where you have a lot of slugs, and then they lived in the inland. There are also inland populations where there are fewer slugs. And he developed a feeding score, a slug feeding score, based on tongue flick rates. And he found that the coastal populations had a much higher slug feeding score than the inland populations. So there's this behavioral preference for what you recognize as food depending on the food that is present where you're at. And so if you're a snake and you live in a place where there are a lot of slugs, you learn to eat slugs and you learn to like it. Now I sound like my mother. You'll eat the food in front of you and you'll like it. Clean your plate. So your dietary preferences get influenced by the prey environment that you find yourself in. So if you find yourself in an area where there are lots of newts, and there are lots of newts in many of these areas where newts occur, you might develop a dietary preference to feed on these newts simply because that's the food that happens to be there. Not surprising that Little Butch started thinking about these things while he was at the University of Chicago, working with Steve Arnold, who was already thinking about what snakes eat and why snakes eat them. Because what he eventually did after he graduated with his PhD is he basically said, I'm going to start tackling this snake and newt issue in a serious way. I want to know how these two species have co-evolved such that we have really, really toxic newts and really, really resistant snakes. And this is a Thamnophis totalis eating a rough-skinned newt. And we're just about practically out of time, so we'll pick this up. We have a few more minutes to spend on this topic. On Monday, we're going to talk about regression, which is one of the primary statistical methods that they use throughout this case study. So over the weekend, part of why I wanted you to read this chapter is because it introduces you to the case study. The other reason that I wanted you to read this book chapter is in the book chapter, Brody really tries to get across the idea that scientists think and behave in certain ways. When they're just out in nature, they have this kind of innate curiosity and they think about problems in particular ways. So what I want to start with on Monday is, what are some of the salient characteristics of scientists that you see in the Brodies as you go through this process, and what do these things say about what it takes to be a successful scientist? And those things should come out of your reading. If in order to answer these two questions you need to reread the chapter over the weekend, by all means reread the chapter. It's not a bad read. Questions? All right, good job today.